Okay, so welcome everybody to session 3 of Inspiring Future STEM Stars. I just realized that this is our third session and I've never really properly introduced myself to all of you. So once again, my name is Hannah. I am in the 11th grade. I've been a Girl Scout for 10 years now. I love STEM. A little bit more about the STEM things that I've done in high school. I've taken honors chemistry, AP biology, and I'm taking a college Java programming class. I also like to do STEM clubs like chemistry, Women in Science and Engineering Club, and coding club. So I am very excited to introduce you to our guest speaker for today, Liza Zanotti. She's an advanced placement biology teacher.
Good morning, everyone.
Hi, Mrs. Zanotti.
Hi, nice to see you, Hannah.
Nice to see you too. Thank you for joining us today.
I appreciate it. I'm very excited to see all these young ladies that are excited to be going into possibly a career in STEM. That makes me so, so happy.
So, Mrs. Zanotti, what do you do for work or research?
So I am a high school biology teacher. I actually went to undergrad. I got my undergrad. I majored in biology with a minor in chemistry. So in addition to being a teacher, I have a biology degree. I did research in cancer, and I did a lot of cool things that I use in my AP bio classes that I teach my students. At the high school, I teach an AP class, and I teach classes that are kind of geared towards the medical field. And there's so many STEM opportunities, even in the medical field. There's so many different things that we do in classes to kind of just make, give the opportunity to students to do things that they could possibly pursue. And maybe it's in robotics, or maybe it's in, research. But yeah, so that's what I pretty much do for the most part. Is I'm a high school teacher with a bio degree and a minor in chemistry.
What's your favorite part about your career?
I think the best part for me is, when I went into school, I've always been really strong in math and science. And when I was young, girls didn't do that that much. And I always thought to myself, well, it doesn't matter who you are. Doesn't matter what your background is. If you have passion and you have love for science, that's what you should pursue. And so that's what I chose to do. And the reason why I love what I do in my career is because I get to help students. So I get to be more than just, one person in a STEM career. I can help a lot of students, a lot of girls go into a career for themselves. And that to me is most impactful, that one person can have impact and help students find passion in a career in the STEM field.
What did you like about STEM, and how did you know that you wanted to pursue it?
Well, I think I've always been a thinker. And so one of the things about STEM that I love, I wouldn't just look at something and say, oh, okay. I would look at, I don't know, even a puzzle and say, how does that work? And I would always want to try to touch it, feel it, move it, try to get to the bottom line of how, why, what's happening here. And that's always been my mind. My mind has always been like that from a little tiny girl. And I just would get excited when I would have science classes. And I was always really excited about math too. And the best part about STEM is that it marries the two together. And so that's the best reason for, the STEM career. And the reason why I pursued it is because of the passion that I had for STEM, and also the passion for science. And to me, science is the most powerful realm of what we look at, because it really looks at the world in a different lens. And so that's why I love what I do.
What was your personal experience pursuing STEM?
Like I said, when I was growing up, It was a little bit difficult sometimes. I had teachers just like myself now in chemistry. I had a chemistry teacher that was in my honors chemistry class, and he said that I was going to be going into the STEM field, and I really wanted that to be the truth. And with his positive outlook, and a couple of my other teachers that had really positive outlooks, I originally wanted to become a nuclear medicine technologist. And that's something that I was going to school for at the very beginning. So it's very different than where I am right now. But the reality is, like I said before, being a teacher, I can impact so many more girls to go into STEM than if I just myself went into it. So.
Okay, so this is a little bit different, but it's also on topic since we're going to be doing DNA extraction today. I know in our AP Bio class, you always mention using PCR and about your experience studying DNA. What can you tell us about that?
Well, really, what's cool about PCR, imagine this, okay? So imagine you have DNA, and DNA is the inner workings of all of the information your body would need. So you open up a box and there's instructions, right? The instructions will tell you, this is how you build whatever toy you got, or this is how you build a new bed that you just had delivered. So DNA is the inner workings of our body to tell us, okay, this is how we build all the things that we need to be an organism, a whole entity, a whole body, right? So PCR is a tool that we can use to look at small amounts of DNA. So imagine that we want to have more of the DNA. I have to make a copy of it, okay? So when you make a copy of it, I could hand write a copy on a piece of paper, right? If I have instructions, I can hand write. But that would take a long time to do. And I wouldn't probably be able to get a lot, right? But what if I instead had a copy machine, a machine that could help me make a little bit of DNA and make it into a lot of DNA? And that's what polymerase chain reaction does. It actually takes your DNA, small fragments, and it builds what you have into a lot of copies of that fragment. And so we usually use PCR at a lot of different reasons. But if for me, I was using a PCR machine to look at cancer and looking at what genes, which genes are what are made up from DNA. So DNA make up genes, right? And those genes is what I was looking at to see, oh, is this particular, is this person going to be more likely to get this type of cancer? So that's what I did with my PCR.
That's really cool. So, what inspired you to pursue your field, and what was it like being a woman in STEM in your field?
I think for me, it's always been just passion. Knowing that from like a young age, there were things that were put in front of me, and I always really enjoyed teaching other people. So when I was in college, I did really well in my science classes, but I also did really well in math. And so when I did really well in math, my math teacher said, hey, do you want to be a TA, which is the teaching assistant of a math class? And I thought, well, wow. I said, why would I do that? Can you explain? So I was going to be paid to go to the class like a normal student. So I would get paid. And then you would have an additional day that I would actually run the training. So I would be the teacher that would teach the information about concepts that people were not understanding. So that's exactly what I did. So that's part of the reason why I am a teacher now, because I really enjoyed helping other people learn the curriculum about things that I'm passionate about, which is science and math.
Do you have any advice for students interested in pursuing STEM?
Yeah, I will say this. STEM is not an easy career. The curriculum is not easy, but anything that you truly have your mind set out to do, you will be able to succeed. I think that you have to have that the drive. You want to make sure that you love science. And so if you have a love and a passion for whatever it is you do in STEM, then you're going to want to do better, learn more, and work harder because you're going to be passionate about what it is that you're doing.
That's fantastic. So does anyone have a question for Mrs. Zanotti? If you have a question, please feel free to unmute yourself and ask your question.
I have a question. Did you ever have another job you wanted to do besides STEM, or was that the only job you ever wanted?
Well, great question. Thank you for asking. I always knew I loved science. I just didn't know what field of STEM I would really want to go into. So I always knew that science was my passion, and I followed my passion. And what's nice about STEM is that there's so many different things you can do. So if you think about something you might be able to do, you could do it, and it's a STEM-based field. But then there's another turn or like a door that opens, and then you have another opportunity that's STEM-based, but it's completely different than the one you originally were looking at. So for me, I never had another job, but I did have originally a different end career path, which, like I said, was to be a nuclear medicine technologist.
Are there any questions? Other questions? If you're writing on the screen, can you please stop? Thank you. Are there any more questions for Mrs. Zanotti? Okay, I don't hear anybody. So thank you very much, Mrs. Zanotti, for coming out here and inspiring all of these wonderful participants. Oh, it looks like we have a question. If you have a question, please feel free to unmute yourself and ask Mrs. Zanotti. Okay, I don't know what that was then. Thank you. Thank you so much for coming out here and inspiring all of our participants. I really appreciate you taking time out of your day to join us.
Thank you. Enjoy your DNA in a little bit. Thank you, guys.
Have a great day.
Yes, you too. Bye-bye.
Bye. Okay, so next we're gonna move on to our Crazy Milk Lab. So the materials that you're going to need are a small dish, such as a petri dish, or a small bowl or plate. You will need milk that has fat content, so try not to use skim milk because it won't work as well. You're also going to need liquid dish soap, food coloring, and a cotton swab. And I'll play this video while you gather your ingredients, if it lets me. So this is a cool video. Somebody did a really large Crazy Milk Lab experiment. So this is just so that you can see it on a really large scale. My apologies. So once you've gathered all of your supplies, we're going to move on to the next step. So for the procedure.
We don't have all our stuff yet.
Okay. I can wait a few minutes for you. No worries. Please feel free to tell me when you're all ready to move on.
I have a question.
What is your question?
Is this a good container for it?
Let me see. I am not sure who I'm looking at right now. Oh, that will work. You just need enough milk to cover the bottom of the container. Okay, So it looks like pretty.
I have a question.
What's your question?
I don't know where my cotton swabs are. Is it okay if I use something else?
Yeah, you could probably use a napkin or maybe a small piece of toilet paper and just put soap on the end of it. And then you're just gonna basically wrap that up so it's kind of, do you have a napkin or something that you could use?
Yeah. Yeah.
Just kind of wrap it so it's kind of in a stick kind of form. Do you know what I'm talking about?
Yep.
Cool. That should work.
Thank you.
No problem. Okay, so I think almost everyone has their supplies ready. So I'm going to move on to the next step. Don't worry if you're behind, you'll have time to catch up. So what you're first going to do is you're going to pour some milk into the bottom of the container, and then you're going to put a drop of each color food coloring near the center of the container, as indicated below by the small diagram that I made. So then you'll put a drop of soap on a Q-tip and put that in the center of the dish. You can also put soap on the other end of the Q-tip and put it in a different part of the dish, or blow on the dish to see how that affects the color movement. So here's a video of me doing the experiment to help clarify things. Let me know if you can hear it. Hi everyone, so today we're going to be doing the Crazy Milk Lab. This is actually a really cool experiment. We did it in my honors chemistry class. So today you'll be needing milk, a petri dish or a shallow plate or bowl, food coloring, liquid dish soap, if straw if you want, and Q-tips. So the first thing that we are going to be doing is we are going to pour a milk into the bowl, just enough to cover the bottom surface of the dish. Okay, next, we're going to put a drop of each color food coloring near the center of the dish. Okay, so once you put all of the food coloring in, get a Q-tip. Make sure that you cover the end of it really well with dish soap. And then let me make sure that you can see it in the camera. We're going to put the Q-tip in the center of the dish. You're going to see all of the colors kind of move away. And then they're going to swirl a bunch. It's really cool. If you want, you can put soap on the other end of the Q-tip and put it in a different spot. For even more fun, you can use a straw and you can blow on it and see how the colors move. Hope you enjoy. Okay, so can anybody share their observations about what they noticed while conducting this experiment?
All the colors, once you put it in, they spread it out. So it was like then they started mixing afterwards.
So it was kind of like when you see something, I don't know, like they all spread it out. So sort of, it's like the soap, the stuff in the soap that made it all spread out.
That's awesome. Does anybody else have something they'd like to share?
When you put the soap of the dish soap on your Q-tip, when you put it down, the color starts spreading out. They start spreading out.
It's pretty cool, isn't it?
Yeah.
It looks like somebody wrote in the chat when the soap was put onto the milk, the color spread out. Yeah, that's pretty much exactly what happened.
I see, Baba, but I gotta clean it up.
Okay, so next we're gonna learn about why this happened. So on the next slide, I'm gonna use kind of big words, but I'm gonna break them down for you right now. So hydro means water. So you can just think of hydro as liquid or water. And phobic is it's a dislike for something. So it's like if you don't like spiders, you want to stay away from them, right? But philic means that you like it. So you don't mind being next to it. So why do these colors move and swirl? That's the big question, isn't it? So there's fat in the milk, and it doesn't break down in water.
Yeah. Why?
So the fat doesn't break down in water, and that's because it's hydrophobic and nonpolar. That means it just doesn't like water. And the dish soap is composed of things called micelles, which are soap molecules that have a hydrophilic head, which means the head likes to be around water, and a tail that's hydrophobic, which means it hates being around water.
And when you
I'm gonna get this thing to true.
So why do the colors move and swirl? We're going to get to that. So the hydrophobic tail, the part that doesn't like water, it likes the fat molecules that are in the milk, while the hydrophilic head, which likes water, it likes to connect to the water molecules in the milk. So basically, the soap molecules are going to basically really quickly move around the dish, and the tails are going to try to connect to fat molecules. And as they're trying to search for more fat molecules in the milk, they're going to bump into the food dye particles, and that causes the colors to move around, and it makes it that tie-dye appearance. It's really cool, isn't it? So these are some research sources that I got for the information. So now we're going to learn about DNA extraction. What is DNA? So as we heard Mrs. Zanotti talking about earlier, it's the genetic blueprint in our cells that basically makes you you. It codes for everything that makes you you. It codes for proteins and all kinds of stuff. It's your genes and all of the genetic material. So it's found in the cell nucleus, which is basically the control center of the cell. And its structure is called a double helix, which is basically like a ladder, except it's twisted. And it has 2 anti-parallel strands. There are also 4 bases: adenine, thymine, cytosine, and guanine. And these combination of bases, how they're ordered and paired, that makes you who you are. And DNA stands for deoxyribonucleic acid, but that's just a big old word that basically means DNA and genetic blueprint. So some cool facts about DNA. Human DNA has about 3 billion base pairs. And all humans have over 99.9% of their DNA in common. It's that 0.1% that makes us all different. DNA can show who you're related to, and DNA is used to help solve crimes. So in this image over here, they can match the DNA sequences. I believe that's called electrophoresis. We learned about it in AP, but I don't remember the word. It also can be used to help match donors to those in need of transplants. You also have DNA in your mitochondria that you inherit maternally. So now we are going to learn about the structure of DNA. And what better fun to do that by making an edible model. So for this little experiment, you're going to need 2 pieces of licorice or Twizzlers, 10 toothpicks, and 20 mini colorful marshmallows. I'll wait a few seconds in case anybody wants to gather those materials if they have them.
How many marshmallows did you say?
20 mini ones, but you can also make a smaller DNA strand if you don't have that many marshmallows. So I'm gonna start the video. It goes pretty slow, so if you're behind, don't worry.
How many toothpicks?
So since we've been learning a lot about need DNA,
10
I thought it'd be really cool if we could make an edible model that shows us the shape and structure of DNA. So to make this model, what you're gonna need are 2 pieces of licorice, 10 toothpicks, and 20 marshmallows. So I have 5 green ones, 5 mini orange ones, 5 pink ones, and 5 yellow ones. So basically, DNA has a really cool structure. It's a double helix. So that's basically like a twisted ladder. And once you finish this model, you'll see exactly what that looks like. So DNA has basically 2 backbones. And these backbones are made out of sugars and phosphates. And that's what the Twizzlers are going to represent. So DNA actually stands for deoxyribonucleic acid. Just a little fun fact. Okay, so we have our 2 phosphate sugar backbones. And what we have, these different colored marshmallows are the different bases. So DNA has these 4 bases that are nitrogenous bases, which means that they contain nitrogen. The bases are adenine, thymine, cytosine, and guanine. And these bases are really kind of cool because they only pair with certain ones. So A always pairs with T, and C always pairs with G. Kind of like apple in the tree, car in the garage. So what we are going to do is we're going to figure out what we want each colored marshmallow to represent. So I'm going to have the green ones represent adenine, the pink ones represent thymine, orange cytosine, and the yellow guanine. So that means the green and the pink are always going to pair together, and the orange and the yellow are always going to pair together, just as how the bases do in DNA. So to make your DNA model, what you're going to do is you're going to put the bases that pair together on the same toothpick, just like so. You're going to kind of sit in the middle, and then you're going to insert the ends of the toothpicks into the licorice backbone, kind of like that. So basically, between just a fun fact, The bond between the two bases is called a hydrogen bond. So next I want C and G to be together. And I'm just going to put them on the Twizzler backbone. Or licorice. So the thing about DNA is that it contains all of the genetic information that makes you you. You inherit it from your parents, and it's found in all of your cells in the cell nucleus, which is kind of like the control center of the cell. It's really cool. So how's everybody's edible DNA models coming?
Good.
Mine is choosing that it's not going to be twistable.
Oh, it might be because the toothpicks are too, well, is it really compact? Like, I don't know how to say this. Did you push the
Sturdy?
well, it might be too sturdy. That could be it.
Mine fell apart.
Well, I'm sorry about that. So, is it possible to rebuild it, or did it just like fall apart?
Like I'm back at the beginning.
I'm sorry. Make sure that you're stabbing the toothpicks all the way through the licorice stick. That might help. Okay. So it looks like almost everybody's ready. So I'm going to move on to the next slide. Okay. So why are we going to be using strawberries for a DNA extraction? So what's really cool about strawberries is that they're octoploid. which means that there are 8 copies of each chromosome. And that means that there is a lot of DNA that we can extract. So to compare to humans, we are diploid. That means that we have 2 copies of each chromosome, while strawberries have 8. And also, strawberries are quite soft, so they're easy to mash into small pieces, which is important for this experiment. So these are the supplies that we're going to be needing for this experiment. 1 to 2 strawberries, dish soap, water, cold isopropyl alcohol, which is rubbing alcohol, salt, a plastic bag that zips, so like a Ziploc bag, a strainer or coffee filter, or you could do a funnel and filter paper. You just need something to strain it, really. A container or 2 glasses, tweezers, a toothpick, or a coffee stirrer, or anything that you want to use to try to pick up the isolated DNA. I'll wait a few minutes while everybody works on gathering the materials. If you have any questions, feel free to ask me. If you have a question, feel free to unmute yourself, and I'll do my best to try to help you.
I don't have. We don't have strawberries.
You don't have strawberries? Okay.
No.
1 second. I can. Do you have maybe bananas?
Yeah.
I'm gonna look up if those work, because I thought that you can, but I'll just double check.
Mom, come here.
Bananas are octoploid. I looked it up.
I'm just seeing if you need to do it differently, but you can extract DNA from them. I know that. It looks like you're gonna need to blend them. The recipe is quite similar, but I'd use just about half a banana because it looks like they're doubling the recipe. If you didn't refrigerate or freeze or put the isopropyl alcohol in your freezer, I recommend doing that because it needs to be cold for the experiment to work.
I don't have the rubbing alcohol, and do you need to cut the tops off of the strawberries?
Yes, you're going to need to cut the tops off the strawberries, and if you don't have rubbing alcohol, this session is going to be recorded and posted on YouTube in case you need to complete it if you want to try it at a different date when you have all the supplies, but I don't think it will work if you don't have rubbing alcohol.
Okay.
Sorry about that.
I need to cut the tops off my strawberries.
Okay, so it sounds like everybody has their stuff, so I'm gonna go to the video.
My strawberries aren't cut.
So make sure that you're putting the isopropyl alcohol in the freezer if you haven't done that already because it needs to be cold. And then to create the extraction mixture, you're going to measure out 6 tablespoons of water and pour it in the glass. Add 2 teaspoons of dish detergent and add 1/4 teaspoon of salt and stir it, but you can add more salt because I think that might help it work better. because when I did it, I didn't get too much DNA isolated.
Can you go back to the first slide?
Yeah, there's also an instructional video, so I'm just gonna quickly go through these, and then you'll see it all again. Is that okay?
Yeah.
Okay. So we're gonna cut the tops off the strawberries. We're gonna put the strawberries in the Ziploc bag and close it. And then we're gonna mash up the strawberries. And then we're gonna pour in the extraction mixture that we made on the previous slide. Then make sure that you remove the air and then mash it all up. And then pour this mixture and strain it so that you get all of the clumps, big pieces of strawberry separated. And then you're going to pour about 1 to 2 teaspoons of cold. It's important that the isopropyl alcohol is cold into the container with the strawberry liquid and extraction mixture. And then you should notice some white things float to the surface, and that's the isolated DNA. So I'm going to move on to the video where I walk you through all of the steps. Hi everyone, so today we're going to be doing a strawberry DNA extraction. So for today, you are going to need coffee filters or a strainer, a glass to mix in with 6 tablespoons of water, Ziploc bags, dish soap, stir sticks, strawberries, a teaspoon, a quarter of a teaspoon measuring, salt, alcohol or rubbing alcohol that is cold, so I recommend putting it in the freezer about 20 minutes prior to completing the experiment.
Wait.
Possibly a knife if you need to cut. Okay, so first things first, we have our glass with 6 tablespoons of water. Okay, once you have measured out your dish soap and put it in the water, you're going to measure out a quarter of a teaspoon of salt. And mix this into the glass. Okay, so once you have all of your ingredients in the extraction mixture, you're going to stir it with your stir stick.
How much soap do we need? 2 teaspoons.
2 teaspoons. Once everything is all stirred up nicely, you're going to open a Ziploc bag. Next, take your strawberries and cut off the top leaves. You can set those aside because you won't be needing them. Okay, so once you have your strawberries, you're going to put them in the bag. And at this point, you can pour in the extraction mixture. Now you're going to make sure that you zip up your Ziploc bag very tightly and make sure that you try to get as much air out as possible. Okay. Once you have made sure that is completely sealed, you're going to mash up your strawberries. You might want to use your hands. If there's too much air, just open your bag and carefully try to get more air out without spilling it. I'm using a cup to roll over the strawberries to try to smash them some more. Try not to have any really large chunks. Once everything is all mashed up, it should look kind of like this. You're gonna get the glass that you want to extract the DNA. Next, you're gonna take your coffee filter. You can use one or two. I'm gonna try just one. So it's a circle. You're gonna fold it so that it's a half circle, and then you're gonna fold it in half again. Now it should be a quarter of a circle. Then you're going to open it so that it's a funnel shaped. And set that in the glass. Next, you're going to pour the strawberry and extraction mixture into the strainer. So now I'm going to store the filtered paper in the Ziploc bag that I use so that it doesn't make a mess.
When should we put in the rubbing alcohol?
That's the last step. I'm about to show that. You kind of get the idea. You're almost mushy there. Parts of the strawberry left in. So the last step is you're going to get your teaspoon. One second, I have to double check the measurement. Pretty sure that it is. Okay, so I say 1 teaspoon, but I found that if you pour more rubbing alcohol in, that you're able to isolate more DNA. So maybe 1 to 2 teaspoons will be better, because I found that that helped significantly. Once you pour that in, you're gonna stir it. You should be seeing white pieces at the top of at the surface of a liquid, and those are the pieces of isolated DNA. Good. So you're gonna start to be seeing some white stuff float to the top, and that's the DNA. So all of those white pieces, they're gonna kind of go up together. I can try to get some out. So that white stuff is the strawberry's DNA. Okay. So was anybody able to see the DNA?
Not yet.
Not yet? I'm sorry, but if you're drawing on the screen, can you please stop? Thank you.
Okay, I'm gonna put you in the bathroom.
I don't see anything.
Just wait. It takes time for reaction. Yeah, I think I see a little bit.
be able to see too much, but if you pour in more of the cold alcohol, it should help a bit. I found that helped me see a lot more of it.
Oh, I see it. It's coming. I see it.
I see it too.
What?
It's saying.
I see
Oh,
it.
she can hear us.
I
Sorry.
can see it. It's awesome. Come on. Here, Mom. Come here.
It should be the white pieces on top of the liquid. Yep. So I have a really cool picture of it, I think, on the next slide. So yeah, these white pieces, those are the strawberry's DNA. It's really cool, right? So I'm gonna explain why we use all of these different things. So the soap, the dish soap, that helped dissolve the fruit cell's membrane. So the cell membrane, it's a phospholipid bilayer, which basically just means that there's these little phospholipids that have a part that like water and a part that don't like water, and then there's just 2 layers of them. And those are basically kind of the skin of the cell. They help provide structure, and they also help with transport. And salt, that helped remove the proteins that were attached to the DNA and help keep them dissolved. And then at the final step, when we poured in the cold isopropyl alcohol, that caused the DNA to precipitate into the solution. That just means that it doesn't break down and that it forms in the solution. So that's how it just magically, well, not magically, it just kind of appeared once we poured that in because it precipitated. Does anybody want to share what they found? Did it work?
It worked for me. I see a lot of like the white pieces.
Same with me. It works for me. We saw a lot. We have lots. I see lots. I see lots. I'm not.
You were a little more listening.
You said to probably be at the top.
See if it makes more.
Yeah, you don't.
You see, it's clear up at the top.
Okay, now we must sign up for DNA extraction at GPO.
That's right, we've done it.
Oh, you have?
No, you just did not. Don and
That I used in case you want to learn more. These slides will be posted on my project's website so that you can access.
Yeah, Daddy, it works. You can see the DNA flakes at the top.
So thank you everybody for participating. I really hope that you enjoyed this 3rd session. My email is inspiringfuturestemstars@gmail.com. And I highly encourage you to email me if there's feedback on how I can make next session more fun. I also really would love it if you all RSVP for our next session on July 29th. We're going to build various mini rockets, like bottle rockets, film canister rockets, and balloon rockets on a string. And my Flipgrid for this project is posted here, and I can also send out the link if you guys want. It has STEM challenges, a virtual science fair where you can post videos of what you want to test, and as well as other STEM challenges like aluminum foil boats and different other challenges. And if you have any questions, I'll stay on the Zoom for a few minutes to answer questions. Thank you.
Thank you.
You're welcome.
Thank you.
You're welcome.